Hello friends, welcome back to my YouTube channel Journey with Vijay Kumar Shrivastav. Today we are going to study the topic Rouging in Hybrid Seed Production of Field Crops. First, to understand what is rouging. Rouging refers to the act of identifying and removing plants with undesirable characteristics from agricultural or seed production fields on visual inspection basis. Rogues are removed from the field to preserve the quality of crop being grown. Rouging is particularly important when growing seed crops to prevent plants with undesirable characteristics from propagating into subsequent generations. Rouging is an important practice in seed production to maintain genetic purity of the seed. Significance of rouging. Rouging at all stages of the crop in the field is an essential requirement to maintain the variety purity as it was at the time of release or notification. Sometimes rogue plants are not distinguishable before flowering. Therefore, rouging should be done as early as blooming starts. Doubtful plants too should be rogued. The rogued plants should be removed from the field immediately after rouging and destroyed, as they may survive for a few days and may spread their pollen and cause contaminations. Now, criteria of rouging. Rouging is done on the basis of various parameters like plant characteristics. As plant height, the rogue plants may be tall or short, dwarf, erect or vigorous nature growth. On the basis of nodes and internodes, stem color and pigmentation, etc. Now leaf characters like leaf color, angle of leaf, erect or droopy leaf nature, narrow or broad, number of leaves, size of leaves, leaf sheath color, and on the basis of arrangement of leaves. Now flowering time. The rouging may be done also on the basis of early or late flowering nature or panicle initiation. On the basis of reproductive and flowering characteristics like flower color, pollen color, size of flower, silk or tassel color, pigmentation, and stigma opening, etc. Then on the basis of maturity, it may be early or late maturity. On the basis of panicle exertion, there may be complete exertion, partial exertion. On the basis of panicle length, panicle girth, color, spikelets, and pigmentation. Then on the basis of seed appearance like seed size, seed color, seed shape, and presence of pigmentation, etc. And other bases like diseased and infested plants should be rouged out. Rouging in hybrid seed production. It should be done timely to rogue out undesired plants as off types, pollen shedders in A line, semi-sterile plants, volunteer plants, other crop plants, and disease heads. For effective rouging of off types, it is necessary to have knowledge on the distinguishing characters of identification of A and R lines. Rouging stages are important. It should be done timely and prior to any contaminations. It is done from vegetative stage to harvesting based on many parameters as morphological and reproductive characters. The critical stages are most important in rouging like vegetative stage, pre-flowering stage, flowering stage, and maturity stage. Rouging on vegetative and pre-flowering stage is based on morphological characters as height, color of vegetation, leaf size, shape, orientation, or diseased or malformed plants. Flowering stage rouging based on flowering behavior, emergence of panicle characters, fruiting body, etc., and uprooting of such plants needed. Flowering stage rouging is equally important in case of hybrid crops, where in a male sterile line A line, a plant of male fertile line which is known as B line is present sometimes. Such B line plants are called as pollen shedders, and it must be rouged out at flowering stage. In male lines, R lines, rouging should be done first and completed before flowering and crossing starts. Maturity stage rouging is important and it leads to removal of defective ear heads like off textured, off colored, diseased or malformed, etc. Now we will go through rogging practices in various crops hybrid seed production. Like first one, cotton hybrid seed production. Rogging should be done timely and prior to any contamination. It is done from vegetative stage to harvesting based on many parameters as morphological and reproductive characteristics. In cotton hybrid seed production, the critical stages are most important as vegetative stage, pre-flowering stage, square formation stage, flowering stage, ball development stage, maturity and picking stage. In cotton HSP fields, first rogging should be done in male block before crossing to avoid any contaminations while pollination. In cotton hybrid seed production, rogging is compulsory in A and R lines, means female and male lines. We have to check any male and female mixing. On the basis of plant height, we have to rog out tall or short plants. On the basis of branching pattern, like plants may be open, erect or bushy type. On the basis of leaf size, leaf color, lobes, hairiness and glabrous nature. On the basis of size of squares, buds and stigma nature. On the basis of flower color, anther and pollen color. Flower pigmentation presence, if any. On the basis of ball size and ball shape and its arrangement on plant. And there should be strictly self balls removals. Rogging in maize or corn hybrid seed production. Based on distinct and diagnostic characteristics furnished by breeders, we have to remove off-type plants, diseased plants, late and weak plants. First rogging should be done at 15 to 20 days crop stage, where we have to remove vigorous plants, remove other undesirable plants like late germinated, diseased, insect pest affected, and remove plants with variable leaf traits. Second rogging should be done before flowering, where there should be removal of plants with variable stem color and growth habits, and remove weak plants and diseased plants. Third rogging should be at flowering before anthesis, where we have to remove late plants, remove plants with variable height, and remove plant with variable tassel, silk and husk traits. And the fourth rogging should be at harvesting, where the rogging is done on the basis of cob size, seed color, seed size, shank color and remove disease affected cobs. Rogging parameters are specific to seed crop and is done from seedling stage to harvesting stage based on the phenotypic characteristics. Remove tall and vigorous growing plants from the knee-high stage onwards, and rigorous rogging should be done to remove off types before they shed pollen in corn seed production. The rogging basis in corn hybrid seed production is on the basis of stem color, plant structure, number of leaves, arrangement of leaves, drooping or erect nature of leaves, auricles, nodal color, tassel color, silk color, sheath color, number of cobs, size of cobs, seed size and color, and shank color, etc. Now, rogging practices in hybrid rice seed production. Although rogging can be done at any time of the crop stage prior to contamination, off types have to be removed from the crop whenever they appear. But the three stages are most important in terms of maintaining high genetic purity in case of hybrid rice seed production. Like first one, vegetative or maximum tillering stage. Second is flowering stage, and third, post flowering or before harvesting. Rogging is done to maintain good quality in rice seed yield to achieve good seed quality value. Any admixture will bring down its quality value. Off types, bee-line plants, other distinguishing varieties, diseased and insect infested plants should be rogged out. In hybrid rice seed production, we have to strictly remove maintainer plants. These are the plants of bee-line, more or less similar appearance as the female plants, but these are 3 to 5 days early flowering nature. Then female line. These are fertile plants having yellow, plumpy and dehiscent anthers compared to white, empty and shriveled ones in female. The rogging parameters in hybrid rice seed production is based on plant height, growth duration, spikelet number, spikelet shape and size, spikelet pigmentation or tip points, leaf color and leaf size, maturity, and other morphological traits of type plants can be identified and removed both in A and R lines. During rogging, we have to remove volunteer plants, which are the plants of previous rice crops. Time of rogging in hybrid rice seed production. Rogging should be done from vegetative to maturity stage to ensure that all possible mixtures are removed before harvesting. At vegetative stage, take note of the plant height and the color of the leaf blade and leaf sheaths. At reproductive stage, check the flag leaf angle and degree of panicle exertion. At ripening stage, look for differences in size, shape and color of grains, seeds, as well as presence or absence of awns. Timely removal of off types is important because it will be very difficult to see them if they are shorter or when the rice plants have lodged. Rogging practices in pearl millet hybrid seed production. Rogging is the process of removal of pollen shedders from A lines and off types from parental lines in seed production. Pollen shedders B lines are male fertile plants in A line with similar morphology. Pollen shedder in A line are results of mutations or mechanical mixtures. Off types are the plants distinctly different in morphological characteristics from those with typical characteristics of the line under production. The off-type plants may arise from mechanical mixture or outcrossing and rarely as mutants. Adequate and timely rogging constitutes the most important operation in seed production. Rogs differing from normal plants in phenotypes should be pulled out and discarded at the earliest possible stage of plant growth before flowering to avoid genetic contamination. In case of pollen shedders, the best time for rogging is morning hours when the A line will have least of the exposure to outcrossing and there is little wind movement. Rogging precautions in millet hybrid seed production. Pollen shedders should be removed before anthrax descends. Removal of pollen shedders should be first in the morning activity and is the important aspect of quality seed production. Off-type plants obviously differing in characteristics such as height, flowering time, color of foliage, leaf size, shape and orientation, hairiness, anthocyanin pigmentation, tillering, panicle size or any other morphological characteristic and diseased plants should be removed before flowering. Rogging at maturity is also necessary to remove off types not distinguishable earlier and contaminants affecting the physical purity of seed. Rogging and sorting out of harvested panicles may be necessary in case of diseased panicles. Rog out off types and volunteer plants before they begin to shed pollen. The rogs must be cut from the base or uprooted to prevent regrowth. Rogging in sorghum hybrid seed production. Rogging is specific to seed crop and is done from seedling stage to harvesting stage based on the phenotypic characters. Off types can be identified through plant height, leaf shape, leaf color, stem pigmentation, plant structure, number of leaves, auricles, nodal color, days to flowering and green color, etc. In female line, during rogging we have to remove off types, wild types, pollen shedders, rogues, partials means semestral plants, volunteer plants, diseased plants, presence of R line, mosaic plants and late and early flowering plants. While in male line we have to remove rogues, presence of any A lines, diseased plants, late and early flowering plants and wild types. Remove all the off types and volunteer plants before they start shedding pollen. The rogged plant must be cut from the bottom or uprooted to prevent regrowth. Rog out other related plants like Johnson grass, Sudan grass, forage plants and plants affected by kernel smut and smut from time to time. Rogging in sunflower hybrid seed production. During roguing, the plants rogued from their vegetative phase to harvesting stage based on plant height, head size, branching habit, number of heads and color of seeds. Generally 2 to 3 roguings are necessary. First roguing should be done at pre-flowering stage and other roguing during flowering stage. Before flowering, remove tall, very early and very late flowering plants, branched plants with multiple heads and diseased plants. During flowering, we have to remove the off-type plants. Sunflower continues to shed viable pollen even after removal from stalks. Therefore, the heads should be thrown on the ground with face downward towards the soil. At maturity, remove off-types, diseased plants, wild sunflower plants, and plants affected by wilt, charcoal rot, blight and other diseases. Roguing in sunflower hybrid seed production. Roguing should be done in both male and female parental lines. Remove the volunteer plants and off-types from both male and female parental line. During flowering period, roguing should be done daily to remove the pollen shedders. Pollen shedders should be removed in the morning hours before the bee activity starts. And the precautions should be taken while roguing, like start roguing before off-types, volunteers and pollen shedders in female rows start shedding pollen. Remove plants with pink or purple colored center in the heads, as the cultivated forms have greenish yellow in the center. Remove plants showing branching and multifloret types. Remove diseased plants and plants which are too early or too late in flowering. Before threshing, remove the heads with white seeds or seeds with prominent white streaks. Roguing in mustard hybrid seed production. Roguing should be done 2 to 3 times before flowering based on characters of parental lines for quality seed production. Roguing should be done from the vegetative phase to harvesting phase. Off-types are removed based on branching type, capsule size and color of the seeds. A special care to remove pollen shedders before flower opening in A lines. A special attention to remove objectionable weeds like Mexican pricky poppy, Ergemon mexicana, which is commonly known as Satyanasi in North India. At least 3 field inspections at pre-flowering, flowering and pre-harvest stages are required to maintain the genetic purity of the mustard. Now roguing in castor hybrid seed production. In case of castor hybrid seed production, field inspection, important parameters involved within their permissible limits for ensuring high standard of genetic purity of various varieties, hybrids and their parental material. A minimum of 3 to 4 field inspections necessary. Important parameters, diagnostic characteristics which form the basis for field inspections at different stages of crop growth are week to 10 days prior to flower initiation in primary raceme, full flowering in primary raceme and a week before first picking and flowering in tertiary order spikes. Roguing in castor hybrid seed production. For all varieties and male parents, 2 rounds of roguings are required. First about 10 days prior to flowering and second one coinciding with flowering. During preliminary roguing operations, look for deviants, if any, for various diagnostic morphological characteristics like stem color, internode type, shape of leaves, bloom type, and remove them before flowering. Immediately after primary spike initiation, examine the seed production plots for number of nodes up to primary raceme, type of internode, expression of sex, proportion of male to female flowers in the spike, branching and other spike characteristics in relation to the parameters specified for the variety and parental lines, and pull out all undesirable plants not conforming to the stipulated standards. Any laxity in roguing operations or delay in the timing of roguing would have adverse effect on the ultimate quality of the seed and hence follow the schedule strictly. During flowering rogging may be done 3 to 5 times in interval of 2 to 3 days. Since the flower initiation extends over a period of 10 to 15 days, carry out second round of rogging for 3 to 5 rounds at interval of 2 to 3 days to avoid any possible leftovers. Remove the plants affected by disease like Phytospora blight and Cercospora leaf spot. So this presentation was all about rogging in hybrid seed production of field crops. Thank you very much. I have given here my YouTube channel details, Journey with Vijay Kumar Shrivastav. Having request, please visit the channel, subscribe it and provide your kind and valuable feedback. Thank you.